Hey guys, welcome to another episode of the Human AI Podcast. Today we're going to talk about anemia, lung imaging, and exercise and health. I'm here with Krok, an AI. It's going to be helping us with these topics. If you're unfamiliar with this podcast, I'm a student here. I don't know the answers to these questions. I just try to gain some insights on these topics with the help of AI. Please take everything that AI says with a pinch of salt and, yeah, do proper science, cross reference. I hope you enjoy the video, nevertheless, and you find the information valuable. So, Grok, let's start with anemia. Give us a definition and tell us about the causes.
Anemia is a condition where you don't have enough healthy red blood cells, or hemoglobin, to carry adequate oxygen to your body's tissues, often leading to fatigue and weakness. It can be caused by blood loss, like from heavy periods, or injury, insufficient red blood cell production due to deficiencies in iron, vitamin B12, or folate, or chronic diseases like kidney disease, or excessive red blood cell destruction, as in sickle cell anemia or thalassemia.
I want to ask you about some kind of keywords that you can clarify for us too. Hematocrit, for instance, mean cell, MCH.
Hematocrit is the percentage of your blood made up of red blood cells, showing how well your blood can carry oxygen. MCH, or mean corpuscular hemoglobin, measures the average amount of hemoglobin in each red blood cell, helping diagnose types of anemia.
I see. Interesting. Okay, and okay.
So,
Explain to us some other concepts such as erythropoiesis, erythropoietin. Explain us the shape of red blood cells, why they have biconcave discs, no nucleus.
erythropoiesis is the process your body uses to make red blood cells, mostly in the bone marrow, starting from stem cells and maturing into functional cells. Erythropoietin, a hormone mainly produced by the kidneys, signals the bone marrow to ramp up red blood cell production when oxygen levels are low. Red blood cells are shaped like biconcave discs, which maximizes their surface area for oxygen transport and lets them deform to squeeze through tiny capillaries. They lack a nucleus to pack in more hemoglobin, the protein that carries oxygen, making them super efficient at their job.
Interesting. Okay, let's talk a bit about blood film now. I want to understand the differences between hematinic deficiency, hemoglobinopathy, hemolysis. Yeah.
A blood film is a thin layer of blood smeared on a slide, examined under a microscope to check red blood cell shape, size, and other features. Hematinic deficiency shows up as abnormal red blood cells due to low levels of iron, vitamin B12, or folate, often causing microcytic or macrocytic anemia. Hemolysis anemia involves genetic defects in hemoglobin structure, like sickle cell disease, leading to misshaped cells visible on the film. Hemolysis appears as damaged or fragmented red blood cells caused by conditions like autoimmune disorders or mechanical stress, indicating the cells are breaking down too fast.
And how does hematinic deficiency relate to like microcytic, macrocytic, hypochromic, anisopoikilocytosis? And how does hemoglobinopathy relate to target and sickle cells? How does hemolysis lead to polychromasia?
Hematinic deficiency ties to microcytic anemia, where iron deficiency makes red blood cells small, often hypochromic, meaning they're pale with less hemoglobin. Macrocytic anemia, from B12 or folate deficiency, shows larger cells. Anisopoikilocytosis describes varied cell shapes and sizes, due to these deficiencies disrupting normal cell formation. Hemolysis anemia leads to target cells, red blood cells with a bullseye appearance because of abnormal hemoglobin causing membrane changes. Hemolysis causes poikilolysosis, irregular cell shapes, as red blood cells get damaged or fragmented from mechanical stress, toxins, or immune attacks, visible as odd shapes on a blood film.
Okay, let's talk about hemoglobin structure now. So we have alpha globin chains, beta globin chains. We've got an iron in each one of the globins.
Hemoglobin is a protein in red blood cells with 4 globin chains, 2 alpha and 2 beta in adults, each holding a heme group with an iron atom at its center that binds oxygen. The iron doesn't sit in the globin itself, but in the heme, allowing hemoglobin to pick up oxygen in the lungs and release it in tissues. This structure, with its precise chain arrangement, ensures efficient oxygen transport and stability, but defects, like in hemoglobinopathies, can mess with its function or shape.
Very good. Okay, tell us more about erythropoietin and erythropoiesis. Where do these things happen? And explain us erythroid series, bone marrow aspirate, and bone marrow trephine too.
Erythropoietin is a hormone mostly made in the kidneys, triggered by low oxygen levels, and it tells your bone marrow to churn out more red blood cells. Erythropoietin, the process of making those red blood cells, happens mainly in the bone marrow, starting with stem cells that turn into erythroid precursors over about a week. The erythroid series refers to the stages of maturing red blood cells, from early proerythroblasts to normoblasts, which lose their nucleus to become reticulocytes and finally mature red blood cells. A bone marrow aspirate is a procedure where a needle pulls out liquid marrow to check the cells, like those in the erythroid series, for abnormalities in number or shape. A bone marrow trephine is a biopsy, taking a small core of marrow tissue to examine the marrow's structure, cell density, and how well erythropoiesis is happening. Both tests help diagnose issues like anemia or marrow disorders.
Okay.
Alright, so we were diving into
Why don't we
erythropoiesis.
talk a bit about symptoms and signs of anemia now? Explain us pallor conjunctivae, skin folds in the hand, chylonychia. Why do these things occur?
Anemia symptoms come from low red blood cell counts or hemoglobin, reducing oxygen delivery to tissues. Pallor, that pale look in the skin, happens because there's less hemoglobin circulating, making tissues less red. Conjunctivae, the inner lining of the eyelids, turn pale for the same reason. Less blood flow and hemoglobin show up clearly in thin membranes. Skin folds in the hand, like the creases in your palm, also lose their reddish hue, looking washed out due to reduced blood volume or hemoglobin. Koilonychia, spoon-shaped nails, often shows up in iron deficiency anemia because iron is crucial for nail bed tissue health, and low levels cause thin, consapped nails. These signs all tie back to the body struggling to oxygenate tissues properly or lacking key nutrients like iron.
Okay, and what are some main causes of anemia? Perhaps, iron deficiency, B12 or folic acid deficiency. Explain us how that would lead to megaloblastic anemia too. Explain us hemoglobinopathies. Explain us bone marrow dysfunction. Explain us hemolysis, hemorrhage.
Iron deficiency leads to anemia by limiting hemoglobin production, causing small, pale red blood cells, known as microcytic anemia. B12 or folic acid deficiency disrupts DNA synthesis, slowing red blood cell maturation, resulting in large, immature cells called megaloblastic anemia, where cells are big but dysfunctional. Hemoglobinopathies, like sickle cell disease or thalassemia, involve genetic defects in hemoglobin, producing abnormal or insufficient red blood cells that impair oxygen transport. Bone marrow dysfunction, from causes like leukemia or aplastic anemia, means the marrow fails to produce enough red blood cells, leading to low counts across the board. Hemolysis is when red blood cells are destroyed too quickly due to autoimmune issues, toxins, or mechanical damage, outpacing the body's ability to replace them. Hemorrhage, or severe blood loss from injury or conditions like ulcers, rapidly depletes red blood cells, causing acute anemia if the loss isn't stopped or replaced.
Okay. And let's talk a bit about categorizing by red cell size. So we have microcytic, macrocytic. We have to think about normocytic.
Red cell size helps categorize anemia. Microcytic anemia, with small red blood cells, often comes from iron deficiency or thalassemia, where hemoglobin production is impaired. Macrocytic anemia, with large red blood cells, is typically caused by B12 or folate deficiency, leading to megaloblastic cells, or sometimes alcohol or liver disease. Normocytic anemia, where red blood cells are normal in size but low in number, points to issues like acute blood loss, hemolysis, chronic disease, or bone marrow failure, where production or survival of cells is disrupted without affecting their size.
Explain us reticulocytes in this, how they're involved.
Reticulocytes are young, immature red blood cells released from the bone marrow, still with some RNA before they fully mature in a day or two. In anemia, their count shows how well the bone marrow is responding. High reticulocyte counts suggest the marrow is cranking out cells to compensate, like in hemolysis or after blood loss, while low counts point to bone marrow failure or deficiencies like iron, B12, or folate, where production is sluggish. Checking reticulocytes helps figure out if anemia is from poor production or rapid cell destruction.
Interesting. Okay. And let's go a bit deeper on problems of synthesis now. So with hematinic deficiency, we might have not enough iron or maybe not enough B12. Question, why would not enough iron showcase microcytic cells, hypochromic cells, and B12 would show macrocytic red cells, right-shifted neutrophils, and megaloblastic anemia? Then, talk to us about bone marrow failure too. Why would we have primary and secondary, and have primary called aplastic anemia, and then secondary called infiltration, dysplastic? Explain what these are.
Iron deficiency causes microcytic, hypochromic cells because iron is needed for hemoglobin synthesis. Without enough, red blood cells become small and pale, lacking sufficient hemoglobin. B12 deficiency leads to macrocytic cells and megaloblastic anemia because B12 is crucial for DNA synthesis, so without it, red blood cell precursors grow large but fail to divide properly, resulting in oversized, immature cells. It also causes right-shifted neutrophils, where white blood cells have extra nuclear lobes due to impaired DNA maturation. Bone marrow failure means the marrow can't produce enough blood cells. Primary failure, called aplastic anemia, happens when the marrow itself is damaged, often from autoimmune issues, radiation, or toxins, leading to low red blood cells, white cells, and platelets. Secondary failure, sometimes termed infiltration, occurs when the marrow is crowded out or suppressed by external factors like leukemia, lymphoma, or fibrosis, which replace or disrupt normal blood cell production. Both result in normocytic anemia, but the cause, marrow damage versus invasion, sets them apart.
Okay. Interesting. Very interesting.
Glad you're finding it interesting. So, we were digging into bone marrow failure, infiltration, and how deficiencies shape red blood cells. Want to keep going with anemia or switch to lung imaging or
So,
exercise and health next?
Is there B vitamins in wheat?
Wheat contains several B vitamins, especially thiamine, B1, riboflavin, B2, niacin, B3, and folate, B9. mostly in the bran and germ of whole wheat.
How about 6 and 12?
I'm assuming you're asking about B vitamins, specifically B6, pyridoxine, and B12, cobalamin, in wheat, since we were just talking about B vitamins. Whole wheat does contain some vitamin B6, mostly in the bran, aiding in metabolism and brain health, but the amount is modest, about 0.3 milligrams per 100 grams. Vitamin B12, however, is naturally absent in wheat and most plant foods, as it's primarily found in animal products like meat, eggs, and dairy. Some fortified wheat products, like certain cereals, may have B12 added, but you'd need to check the label.
Okay.
If you
Let's talk about iron deficiency a bit more. So, this might be due to diet. And to confirm it, we might explain me these words maybe. Ferritin, how do we measure ferritin? What's ferritin? What do you mean by serum? What's the word serum in serum iron? What is transferrin? What's transferrin saturation?
Iron deficiency often stems from a diet low in iron-rich foods like red meat, leafy greens, or fortified cereals, or from poor absorption or blood loss. Ferritin is a protein that stores iron in your body, mostly in the liver, spleen, and bone marrow. Low ferritin levels signal iron deficiency. We measure ferritin through a blood test, checking its concentration in nanograms per milliliter to gauge iron stores. Serum refers to the liquid part of blood, minus the cells and clotting factors. So serum iron is the amount of iron circulating in this liquid, measured via blood test. Transferrin is a protein that transports iron in the blood to where it's needed, like the bone marrow for red blood cell production. Transferrin saturation is the percentage of transferrin bound to iron, calculated by dividing serum iron by total iron binding capacity, which reflects transferrin levels. Low saturation, like under 20%, often confirms iron deficiency. These tests together paint a picture of your body's iron status.
Mm-hmm. Okay.
All right, so we were unpacking iron deficiency, ferritin, transferrin, and how those blood tests work. Want to dive deeper into anemia? Maybe explore treatments for iron deficiency, or shift to lung imaging or exercise and health for the podcast?
I want to understand in iron deficiency the different cells. Pencil cells, target cells.
Iron deficiency impacts red blood cell development and appearance, starting with stem cells in the bone marrow. Hematopoietic stem cells kick off erythropoiesis, but without enough iron, hemoglobin synthesis falters, leading to smaller, paler red blood cells, microcytic and hypochromic, seen in later stages like normoblasts and mature red blood cells. Target cells, which look like bullseyes with hemoglobin pooled in the center, aren't typically caused by iron deficiency alone. They're more common in hemoglobinopathies, like thalassemia, where iron is present but hemoglobin structure is faulty. Though severe iron deficiency can occasionally contribute by altering cell membranes. Iron deficiency mainly disrupts the erythroid series, reducing cell size and hemoglobin content, not producing target cells directly. Instead, you'd see more anisopoikilocytous, varied cell shapes and sizes on a blood film due to impaired production. If you're asking about other specific cells, could you clarify?
No, that's good. Okay, I want to ask about thalassemia now, how it connects to microcytic anemia. Explain beta thalassemia, explain the thal trait and the major, thalassemia trait, thalassemia major.
Thalassemia is a genetic disorder causing defective hemoglobin production, leading to microcytic anemia, where red blood cells are small due to low hemoglobin content. It connects to microcytic anemia because mutations in globin genes reduce functional hemoglobin, similar to iron deficiency, but here it's about faulty globin chains, not iron. Beta thalassemia specifically involves mutations in the beta globin gene, disrupting beta chain production for hemoglobin. The thalassemia trait, or beta thalassemia minor, means you carry one mutated gene, causing mild microcytic anemia with minimal symptoms, often just fatigue, and target cells or basophilic stippling on a blood film. Beta thalassemia major, or Cooley's anemia, involves two mutated genes, severely limiting beta globin production, leading to profound anemia, ineffective erythropoiesis, and symptoms like pallor, jaundice, and bone deformities from marrow overactivity. Patients often need regular transfusions and show lots of abnormal cells, like target cells, on blood films. The trait is a carrier state. Major is life-threatening without treatment.
Okay, now I want to ask about pernicious anemia. What's pernicious anemia? What does it have to do with B12 deficiency? And how does it connect with intrinsic factor, with Crohn's and celiac disease?
Pernicious anemia is a type of megaloblastic anemia caused by vitamin B12 deficiency, specifically due to a lack of intrinsic factor, a protein made by stomach cells that helps absorb B12 in the small intestine. Without intrinsic factor, often because of autoimmune destruction of the stomach lining, B12 can't be absorbed, leading to large, immature red blood cells and symptoms like fatigue, pallor, and neurological issues. Crohn's disease, which inflames the small intestine, can damage the area where B12 and intrinsic factor bind, reducing absorption and potentially causing B12 deficiency, though not always pernicious anemia, unless intrinsic factor is also impaired. Celiac disease, by damaging the small intestine's lining through gluten intolerance, can also hinder B12 absorption, leading to deficiency. But again, it's not true pernicious anemia unless intrinsic factor production is affected, like from associated autoimmune gastritis. Both diseases can mimic or worsen B12 deficiency symptoms, but typically involve different mechanisms than the autoimmune loss of intrinsic factor and classic pernicious anemia.
Okay, what about macrocytosis? How do things like alcohol, liver disease, myelodysplasia, pregnancy, can these things cause macrocytosis?
Microcytosis means red blood cells are smaller than normal, often tied to anemia. Alcohol can cause microcytosis by
I was
disrupting...
saying macro, not micro.
Got it. My bad. Let's talk macrocytosis, where red blood cells are larger than normal. Alcohol can cause macrocytosis by directly messing with bone marrow, slowing red blood cell maturation, and often lowering folate levels, leading to big, immature cells. Liver disease, like cirrhosis, causes macrocytosis because it disrupts lipid metabolism, altering red blood cell membranes and impairs folate or B12 storage. Plus, alcohol is often a factor here too. Myeloid disease, or myelodysplastic syndromes, leads to macrocytosis when defective bone marrow produces oversized, poorly formed red blood cells due to genetic mutations. Pregnancy can cause mild macrocytosis because of increased folate and B12 demand for fetal growth, sometimes outpacing supply, slowing cell division and leaving larger cells. All these mess with DNA synthesis or cell membrane structure, pumping out bigger red blood cells seen on a blood film. Want to dig deeper into any of these?